for our second seminar today. This is our second seminar today will be presented by Dr. John Freunds, again, director of the Southern California Particle Center and Supersite. And just to remind everyone out there also that this is being webcast and questions can be sent in, emailed in as well, and we'll have a questions session at the end. Dr. Freunds received his bachelor's degree from UC Berkeley, master's in his doctoral degrees in chemistry from Yale University. I almost said Harvard there. I just kind of flinched, but excuse me. That since he's held several positions, including, I guess, deputy director at NIOSH for a time, and has been at UCLA since 1981. Through that position in the Department of Public Health, the Department of Public Health, Environmental Health, that his research interests have focused on the health effects of air pollutants, toxic air pollutants, and especially particulate matter right now. He'll be discussing really the toxicological implications of some of the research being done by the Particle Center. And also to point out that Dr. Freund serves as chair of the Scientific Review Panel on Toxic Air Contaminants that advises the Air Resources Board, and is one of the original members of, actually the only original member of the Scientific Review Panel. And with that, John, I'll let you begin.
Thank you, Rich. Yeah, I had a birthday the other day, and so these lines about the original member of anything is beginning to get, it makes me nervous. Somebody once said recently I was distinguished, and I also took that as a sign of he was saying that Freund is getting old. Anyway, so I have a lot of slides, and I'm going to cover a lot of material, and hopefully in a relatively reasonable period of time. I wanted to say at the outset that what I'm going to talk about reflects my training, insofar as I think that lots of times when we're talking about air pollution, we talk about chemicals, ozone, NO2, carbon monoxide, so on and so forth. But sometimes we don't do very well talking about the chemistry associated with those chemicals. And so that's where I'm coming from. And so of all the 5 particle centers, ours is the only one where you have a director who's actually a chemist. For those of you who are chemists in the room, at least we have one. And so it does give a slightly different perspective, and I hope I can illustrate that. I was watching Kosis when he gave his talk, and he was clearly looking at this screen because it's the way, rather than at the board. So I won't look at you. I'll look at this screen to some extent. So I apologize for that at the outset. All I want to do with this first slide is simply to show you the participants in this particle center. And as you can see, they're very wide-ranging and cover a wide range of institutions, including Michigan State, where we've done some terrific work, and even the University of Tsukuba in Japan. And I want to add one thing to the funding source, which is that the Air Quality Management District from Southern California is now supporting us. I just want to mention 3 points here. The first is that the principal theme of our research center is mobile source pollution and the health effects associated with that. Within that particular context, I want to focus on our 3 themes, one of which is the investigation of biological mechanisms of particulate matter effects. So biological mechanisms is one of our sub-themes. The second is studies of emission sources and associated adverse health effects. And the third is studies of the effects of varying spatial and temporal patterns of ambient PM and co-pollutants and the resulting health effects, with some emphasis on atmospheric chemistry, and Dr. Ciotas spoke at great length about that this morning. The one thing I want to say in the context of these 3 sub-themes and our overall theme is that they were made possible to some extent by 2 things, one of which was the ARB support for the building of our particle concentrators. Everything I'm going to tell you really wouldn't have been possible without that. And so the ARB should take a great deal of credit for the vision to support the development of the concentrators. And the second thing I want to mention in that regard is that every one of the projects on the list, and I'm not going to go through all the lists, have associated with it an exposure assessment element, a PM characterization part of it, so that what we have tried to do, much of it through COSIS's efforts, has been to, in every study we do, to have it linked with exposure assessment and PM characterization. So this is the list of projects that are underway. I won't go through them. However, when we were reviewed by our external advisory committee, the chair of that committee, Jonathan Salmon, asked me, "Is this your center?" That was a nasty question, if you understand it, because what he was really saying is that, is your center just this list of individual projects? So I said, the heck with them. I'll develop a new graphic that will show what our center is about, and that's that. So this is what I call our horrendogram, and I'm not going to go through this either. I want to say two things about it. First, as you can see, we work very hard at integration across disciplines. And so that's point one. The second point I wanted to emphasize for this audience is, if you notice the two main pictures, that is Freeway study and source receptor study, those are the two studies that are funded by the ARB. So I think it's important to realize that this entire center, which is approximately a $20 million center, that the primary foci, the central part of it, was created by ARB, and in that respect, I think it's terrific. All right, so on to business and talk about science. This is very difficult, I think. What I'm going to do is to focus on part of the set of hypotheses, namely reactive compounds going through xenobiotic metabolism to redox cycling to oxidative stress to this box, which I'll come back to in a second, finally to vascular effects. And I want to say that we've become interested in two other, and they're part of the center now, two other health endpoints, one of which is neurologic outcomes, and the second is reproductive outcomes. So this slide does not convey the health outcomes that we are currently involved in. I do want to mention one thing, is that in terms of asthma and cardiovascular effects, we think that oxidative stress produces a process or a range of responses which begins with an antioxidant response in the lung, is almost simultaneously coupled with inflammatory processes, and finally leads to cytotoxicity, apoptosis, and cellular necrosis. And so when you think about what I'm talking about, think about the fact that we're thinking of these health endpoints as being dictated by a biological process that is relatively complex in its nature. Now, I'm going to show you some things about some of the mitochondrial work we've done, although I won't go into the mechanistic work today. We are also doing research on receptor activation, namely capsaicin receptors, as you can see here. And I won't have time to talk about that. This is another seminar. And we're also particularly interested in covalent bond formation as opposed to redox cycling. And I'll just mention that, but won't really say anything about it. So these elements about the mitochondria, the capsaicin receptors, and stoichiometric chemistry with electrophiles is something that we'll have to talk about at a later date. Now, let me go back and just emphasize that a principal mechanism of our center is that reactive compounds undergo a formation of reactive oxygen species which generates oxidative stress, and I'm going to talk about that for the next few minutes. In terms of that, I just want to mention that it's our general hypothesis that particulate matter can react with cellular electrons from reducing sources to produce PM with negative charges. Negative charge, the PM, can then catalyze the oxidation, the reduction of oxygen to produce superoxide radical, which then goes through the whole series of chemistry that you're aware of, producing hydrogen peroxide and hydroxyl radical. But that basically it's our view that particles are capable of acting as catalysts for the reduction of oxygen. And that's central to this entire point, that we believe that particles, PM matter, is capable of acting catalytically to reduce oxygen and produce oxidative stress. Now, here are some of the ideas that I want to spend a little time on. One, toxicity of PM is due to chemically reactive components, which include metals and organics. However, we now believe that those chemicals don't have to come off the particles to exert their toxicity, and I'm going to talk a lot about that. Secondly, that PM and their components catalyze electron transfer between reducing agents and oxygen to generate reactive species. That's what I just said. Third, that PM is self-contained catalytic entity capable of toxic chemistry when in proximity to metabolizing and respiratory cells. Fourth, that the catalytic activity can amplify exposure effects. 5, that PM may be more active than its components, and I'll come back to this electrochemically active issue. So it's our view, and this is a difficult slide to see, but if you look at a particle that has, say, a copper ion and a quinone, and it picks up an electron to give you a copper 1 ion and a semiquinone, that semiquinone oxygen to produce secondary superoxide and gives you back the quinone. And then, of course, if you can pick up another electron, you can go back to the semiquinone, and this process starts over again. So it is the proximity of metals and organics on PM that we think is important and can lead to the catalytic activity and perhaps the synergism of toxicity associated with particulate matter. And just to make sure that everybody's on the same page, this is naphthoquinone, 1,2-naphthoquinone, that can be can go to the semiquinone, can go to the catechol, and of course, when it forms the semiquinone, the semiquinone can then reduce oxygen to superoxide back to the quinone, and this whole process is catalytic insofar as it's a redox cycling process. And so one molecule of a naphthoquinone can generate tens of thousands of molecules of superoxide. So that the important issue that we're concerned about is when you think of chemistry and you think of stoichiometric chemistry, that is, when you think of acrolein reacting with DNA to form a DNA adduct, that's a one-to-one relationship. So when the acrolein is used up, you've got the DNA, but the process is essentially over. I'm oversimplifying clearly because it's generated in the atmosphere and so on and so forth. But the point I'm trying to make here is we started out our research 4 years ago looking for chemicals that can act greater than the sum of their parts. And that is that they can act catalytically. So we look for chemicals that could be regenerated and that could produce toxic materials as a result. And so our emphasis has been on looking for those kinds of compounds. Nitro PAHs can do the same thing. We're not suggesting that only quinones are active in that regard, but they are certainly a very good model compound for this kind of research. Looking down below, you'll see the 1,2-naphthoquinone reacts with a protein, undergoes a Michael addition, and you see a stoichiometric chemical process. We are also measuring those processes as well, and I won't say much more about it, but we also are developing assays to measure stoichiometric electrophilic reactions as well as redox cycling. Just to And now I want to shift gears for a second and take a little tangent, because one of the reasons I want to talk was because of Jeanette and other people from Stationary Sorts, and because we're interested in talking about what chemicals, based on our research, might be useful to put into the TAC process. And so I want to talk a little bit about naphthalene. But just to go back and remind you of the slide Costa showed you earlier, this is the 12 Children's Health Center health study communities, noticing again a west-to-east pattern. And let's look at naphthalene that we've measured across the Los Angeles basin. In Lompoc, we find 58 nanograms per cubic meter going up to Riverside, and as you can see, there's a very clear pattern that the concentration of naphthalene increases as one goes across the basin. And in Riverside, we've measured values as high as 6,000 nanograms per cubic meter. So, story is there's a lot of naphthalene. The amount of benzoate-pyrene as a PAH is about 0.2 to 0.3 nanograms per cubic meter. So we're talking about a very different concentration, and the question then becomes, is naphthalene an issue of concern, and in particular, is naphthoquinone an issue of concern? And with respect to naphthalene, there is clear evidence of central nervous system tumors. Here in this data, there are nasal cavity adenomas and neuroblastomas. In male rats, NTP says there's clear evidence. In female rats, there's clear evidence. And so there is very strong animal data about the carcinogenicity in rats and mice with respect to naphthalene. And given its concentrations in the Los Angeles basin, this data has real significance. Now, I want to emphasize that neuroblastomas and nasal adenomas have not been observed in chamber controls, and they have not been observed in historical controls. So that what you have is National Toxicology Program have never seen these cancers that we are seeing from naphthalene. And so the National Toxicology Board of Scientific Counselors voted unanimously to list as a carcinogen. Now, I want to take a minute here and just talk a little bit of chemistry. This pathway, this is, let's assume this is benzoate-pyrene or naphthalene. And here you see the formation that every toxicology student sees in the initial formation of an epoxide, which opens up to give you a dihydrodiol by P450 processes, and then you get the diol epoxide, and that results in DNA addicts. However, there is another pathway, metabolic pathway, which uses the enzyme dihydrodiol dehydrogenase, which produces a catechol, which can then result in the formation of a semiquinone and the, in this case, say a benzoate-pyrene compound. The reason I'm taking time to show you this is that not only do we find naphthoquinones in the atmosphere in Los Angeles, but one can metabolize naphthalene via this pathway to the naphthoquinones. And that's important. Whoops. Naphthoquinones produce a mutational spectra in animals that show mutations of the GT transversion variety in P53 tumor suppressor genes. That's important because that's the kind of mutational spectra that you see in lung cancer. This pathway, which is what half the people in this room who've taken toxicology learn is the dominant pathway, namely through the diol epoxide, does not produce the same mutational spectra in lung cancer in the P53 gene. In other words, this pathway explains the lung cancer that we see in many cases in humans better than the traditional pathway that we generally think about. The point of all this is that suggests that we need to do considerable more work on these quinones because they possibly represent certain carcinogens in that respect. And again, if you think of naphthalene, if you're finding 6,000 nanograms in Riverside, and if only 20% of that metabolizes via this pathway, that gives you a microgram, which is obviously a significant amount of material. NPQ is genotoxic, phenanthroquinone is genotoxic, and so on and so forth. And as I just said, 2-dione mutates p53 under redox cycling conditions. That is, that the reactive oxygen produced from the naphthalene produces the genetic spectrum that I'm talking about. So what I want to argue here is that these compounds are really quite important for us to follow up on in the context of the 1807 process. Now, going on to the particle center research a little bit more, just as a background, this shows you the spatial distribution of phenanthroquinone, and as you can see in Mira Loma, San Dimas, Riverside, and Upland on the right-hand side of the chart, that the largest concentrations of the phenanthroquinone that we're finding in the basin is in the eastern region. And I would tend to argue, and I'll ask Kostas to speak to this later, but I would argue that we may be seeing atmospheric chemistry generating those quinones as particles move across the basin. All right, we needed a way to measure redox activity on a quantitative basis because we often don't have good quantitative measures of toxicity. We measure qualitative responses. We don't have a way to measure quantitative responses. So we have developed a technique for measuring quantitatively redox activity. And this is the way we do it. We take a quinone, as you can see here, but in your own mind, replace this with particles, fine, ultrafine, coarse. It doesn't matter. This is, as you can see in the equation, the quinone nor the PM is in the equation. In this case, the quinone is the catalyst. So what we do is we take particles, we put them in a medium with this dithiol-3-etol. It generates reactive oxygen, and so we can then measure the relative toxicity on a quantitative basis of various particles or chemistry in terms of their quantitative ability to produce reactive oxygen. So this DTT assay is a remarkable advance in this field, I think, and that it gives us a basis to measure quantitatively the formation of reactive oxygen. So, looking at some model quinones, you can see that phenanthroquinone, in this case, is the most potent in terms of redox activity, and 1,2-naphthalquinone and 1,4-naphthalquinone also produce it. I took out the slide showing the benzoapyrene quinone activity because it's more of the same. But every quinone that we've tested so far shows redox activity with a couple of exceptions that I'll come back to later. And so, sure.
That the mutagenicity of a lot of the mutagenicity in the Riverside air was due to quinones, wasn't that?
No, they showed that the mutagenicity is primarily nitro pHs, not the quinones. But interestingly enough, Janet would has been asking me if we would support her doing research because she thinks that the nitronaphthalene under atmospheric conditions will produce naphthalquinone. So you have naphthalene going to nitronaphthalene going to naphthalquinone.
Yeah, I managed that research, so just to add to that, in Riverside they found that most of the mutagenicity of the vapor phase was due to nitronaphthalenes, and in the particle phase they found that most was due to nitrolactone of phenanthroquinone primarily.
Right. And the lactone he's talking about was very potent. The problem that we still have a lot of work to do because Janet and Roger do much of their research in chamber studies. And if you ask them how much of what you see in your chamber studies do you find in the ambient environment, they don't really know. And so it's a challenge to go from the chamber-based studies to the actual field studies. And so, for example, the metabolism of naphthalene in the chamber produces some very hot compounds, but to the degree that they have any staying power in the environment is really a good question. Okay, now this is an extremely important slide, and it relates to what Kostas talked about this morning. Namely, this is a series of studies that we've done in which we've been measuring redox activity in particles across the basin. The first slide shows Boyle Heights. It's a freeway, and I'll show you later where it's located, but basically we are here 50 meters from a freeway or 150 meters from a freeway. And for those of you who were in the seminar this morning, you'll know that at 50 meters there's a significant number concentration of ultrafines, and at 150 there's a significantly less. But so we were looking at the redox activity here close to the freeway, and using the concentrators that Kostas developed, we were able to separate coarse, fine plus ultrafine, and ultrafine particles. And so you can look and see the redox activity of the coarse, fine, and ultrafine particles in this slide. And so you can see that, and this is DTT activity per microgram, so it's on a per microgram basis. So you can see that in every case, the ultrafine fraction is the most potent in terms of the generation of redox activity, that is oxidative stress. So that the ultrafines appear to be the most potent by far of the three size ranges. In Claremont, you see the same thing. Again, it's in the ultrafine fraction that we see the greatest potency, and at USC we see the greatest potency. Now, if you look over here at the numbers on the right, and I'm sorry I'm having trouble with this mouse, you can see that in terms of the DTT activity, we find the greatest activity at Claremont, followed by Boyle Heights at 50 meters from the freeway, followed by further from the freeway, BH2, and the USC source. And we would attribute this higher Claremont activity, and remember, we're comparing 137 to 115, so the numbers don't knock you over. But we would argue, and we'll see how this turns out over time as we get more samples, but we would argue that there may be greater redox activity in particles as they undergo atmospheric chemistry and cross the basin. And so it's not surprising to us that we see at Claremont the greatest redox activity in particles. I don't think I'll talk about this, but maybe I'll take one second. If you measure things in terms of not so much the DTT per microgram, but rather DTT where you take into consideration the airborne concentrations, and you look at it in terms of DTT versus per meter cubed, you get a somewhat different answer, because here you're measuring exposure, you're not measuring potency. And you'll notice here that we tend to see the greatest overall redox activity at the freeway sites. Now, the next two slides show you the correlations that we've done. This one looks at OC here, and the R squared is 0.78, and here is OC, and it's 0.72. And so you can see there's a quite good correlation between the redox activity as measured by DTT and the organic and elemental carbon. And also when we look at BGP, which is a measure really, it's a PAH, it's really a measure of gasoline emissions, vehicle emissions, we get an R squared of 0.84. So that, and with the PAHs, it's down around 0.63, but we have a multiplier which is driving that R squared value. But leaving that aside, we see a very good correlation between redox activity in our particles and elemental carbon, organic, and BGP. In other words, that we would argue that based on these results that the redox activity that we see in particles is primarily associated with organic compounds. We see no correlation with metals whatsoever. We think metals play a role because it's all part of the electron transfer process. But based on the evidence that we have, we don't see a predominant effect of metals with respect to redox activity and oxidative stress. When we do the same experiment in a biological study where we're looking at something called heme oxygenase, which is an antioxidant response element, is generated by an antioxidant response element where we're looking at antioxidant defenses. Again, in a biological system, we can see again that the ultrafines continue to be the most potent by far of the three size ranges. And when we plot redox activity that I've been talking about as measured by DTT in relation to the biological measure of oxidative stress, we find a very strong. When we take out this point up here, the correlation is still 0.81, so it's still relatively strong. So that everything that we've done up to now suggests that particles throughout the Los Angeles basin have significant redox activity, and that redox activity is associated with organic chemicals. So overall, there's a strong correlation between particle size, chemical composition, reactive oxygen generating capacity, and cellular oxidative stress. Ultrafine PM is the most potent on a per microgram basis. Now this is an interesting experiment. Here we took some carbon paste and we mixed it with diesel particles, and if you'll notice down here, when we apply a potential across the carbon paste, we basically get no current. But look what happens when we apply a voltage across the carbon paste mixed with diesel particles. We get an enormous current, and this is really an electrochemical demonstration of oxygen reduction occurring. So this is an electrochemical process to show the same thing I've been talking about in the previous slides. And again, when we measure using a Clark-type electrode, when we actually measure oxygen consumption, and I won't go through all this, but these are different reducing agents, and these reducing agents demonstrate that in this case we're using diesel particles, but this data demonstrates that diesel particles are capable of catalyzing oxygen reduction in the presence of reducing agents. So this is a confirmatory study showing that in an electrochemical process that we can reduce oxygen quite literally. So that the data is consistent across three different approaches. So that with that, the bottom line, of course, is that we have to take seriously the mechanism that says redox activity may be a major contributor to health effects related to particle exposure. Now, we've also carried this further, and we've looked at some yeast. One of the good things about yeast is that you can run them in aerobic and anaerobic conditions. So we've looked at the rate of growth of yeast in aerobic and anaerobic conditions, and as you might expect, if you run it under anaerobic conditions in the presence of particles or quinones, you would predict that since you have no oxygen, you can't create oxidative stress. Therefore, your measure of the impact on yeast growth would be different. Namely that we measure something called an IC50, which is the concentration of the compound of interest that will reduce the rate of growth of the yeast by 50%. Okay? And so, if you'll notice here, let's just take phenanthroquinone, for example. If we run it, the micromolar conditions for phenanthroquinone to have it for the IC50 is 14.3, but if we take out the oxygen, it goes to 36.4. That is, the toxicity decreases because the redox pathway is no longer available to impact the yeast growth. If we then take out something called superoxide dismutase, which is an enzyme which destroys reactive oxygen species, you can see that, in fact, the toxicity goes dramatically up, and that so this is consistent with toxicity occurring via a reactive oxygen pathway. So we think that yeast are actually an interesting tool because you could use yeast with various genetic changes. You can use them in aerobic and anaerobic conditions. This is a technique, and I'll just take 1 second on this. We're interested in things like acrolein and crotonaldehyde and other compounds and particles themselves. So we've developed a technique using a molecule, a dansyl nucleophile, and when this molecule reacts with acrolein or other aldehydes, we can form products which we can then measure their formation in a fluorescent assay, so that this is a new technique that we're about to couple with the DTT assay, which will give us a quantitative measure of stoichiometric processes to measure electrophilic chemistry as opposed to redox chemistry. We have also been working on, and I'm going to go through this very fast, we have also been interested in other mechanistic techniques, and so we've been looking at smooth muscle preparations, namely rat aortas and guinea pig trachea, and we've looked at the effects of quinones on tracheal rings, and we find that in fact these quinones, and now we'll look at them with particles, not simply model quinones, but that we can elicit a contraction of the tracheal ring via the quinones. And the 9,10 phenanthroquinone appears to act through reactive oxygen, but the 1,10 naphthalquinone does not. And this is where we are now looking into these capsaicin receptor mechanisms, and I won't say anything more about that. I'll talk about that sometime in the future. But we think that the capsaicin receptor is an important mechanistic element in the toxicities that we're interested in. And Kostas mentioned dicarboxylic acids in his talk today. And so we're interested in products of atmospheric chemistry, namely dicarboxylic acids, as potential molecules that may activate the capsaicin receptor. Okay, we've been interested in exposure models. That is, when you have a particle that deposits in the lung, for example, what happens? And so we, in our simple-minded way, thought about this issue, and we thought of how can we develop a series of alternatives. So we thought that one way toxicity can occur is that the chemicals dissolve off the particle, they then enter cells, and in the cells they generate reactive oxygen. Or you can have absorption of the particle to the cell, followed by dissolution, and then the chemicals enter the cell and generate reactive oxygen. Third is phagocytosis, where the particle absorbs to the cell, is taken up by the cell, and then generates reactive oxygen within the cell. And finally, adsorption and reaction, namely that you generate reactive oxygen outside the cell, it enters the cell and creates toxicity association with redox processes. I'm sure those of you in the room can think of other alternatives to this kind of simple-minded dosimetry, but we're trying to think of things that we could look at. that got us into looking at trying to learn more about what happens to ultrafine particles. Michael Brauer from British Columbia has reported that lungs from Mexico City contain numerous chain aggregated masses of ultrafine carbonaceous spheres, and that long-term exposure to ambient particles, especially to aggregated ambient ultrafine particle combustion products, results in higher retention of these particles, both fine and ultrafine in lung tissue. So we thought that was an interesting paper. And then Andre Nell and our research group started to look at mitochondria, and some of you have seen these slides, and others have read our paper. This is an untreated macrophage, and you can see the sort of biological textbook mitochondria here. You see what happens when we expose the cell to coarse particles. They are taken up and they are isolated in a vacuole, and we see no particles in the mitochondria. When we use ultrafines, however, you see the particles are located all throughout the mitochondria. All throughout the mitochondria. We see particles in the mitochondria we see fine particles in the mitochondria as well, but there's a dose-response relationship that the larger the particle, the less likely we will find them in the mitochondria. But this is the first paper, I believe, that demonstrates quite clearly that we can actually see particle uptake not only into cells, but particle uptake into the mitochondria. And of course, the result of that is destruction of the mitochondrial membrane, and followed by apoptosis of the cell death. And these slides I'm showing you here are B2B cells, which are human epithelial cells that have been transformed, and so they represent the kinds of effects that one would expect to see in human epithelial cells. So this is a nice demonstration, and Andre's continuing this work to look precisely at the mechanism of how the particles get into the mitochondria, and secondly, what is the biology and chemistry that occurs once they enter the mitochondrial space. Okay, I want to shift and talk about some studies that we've done associated with, that's ARB funded, associated with animal studies using concentrated particles. And just as a reminder, asthma is a complex process that involves sensitization, inflammation, and hyperreactivity, to name at least 3 phenotypes. And what I want to show you in this picture, I hate people who show pictures of complicated diagrams in seminars, but what I want to show here simply is interleukin 13 and interleukin 4 as 2 cytokines that bind with B cells that result in the activation of the B cell, and which stimulate the production of IgE antibodies and IgG1 antibodies. And so that interleukin 13 and interleukin 4 are important cytokines within the context of developing asthma health outcomes. And of course, the mast cell plays an important role insofar as it generates proteins that cause airway damage and so on and so forth. But the point I want to make here is that we think that interleukin 4 and interleukin 13 play a particular role with Th2 cells and with B cells in terms of activation and production of antibodies. Secondly, I wanted to mention interleukin 5. Interleukin 5 is a cytokine that results in the differentiation of eosinophils, and those eosinophils produce proteins which then ultimately end up producing airway injury. So we've been interested in terms of our animal studies as to measure interleukin-13 and interleukin-5, among other endpoints, as well as immunoglobulins as well. These two pictures are taken from a review in the New England Journal of Medicine on asthma that I would recommend to anybody who would like a modern update on the mechanisms of asthma. Well, going back to our reactive oxygen hypothesis, we would argue that, in this case, I used diesel particles. I should have changed it to PM, but that PM generates reactive oxygen, produces oxidative stress. Oxidative stress initiates pro-inflammatory effects in target cells, and basically that inflammation is responsible for the adjuvant effects of PM and DEP. Within the context of our freeway study, we would argue that mobile source emissions will exacerbate airway inflammation and allergic airway disease and produce cardiopulmonary effects. Second, that the magnitude of the airway disease and cardiovascular effects are a function of the size distribution. And third, that exposure to ultrafine particles will result in the most severe effects. So we sensitize mice with ovalbumin, and then we expose them to concentrated ultrafine and fine plus ultrafine at progressively increasing distances down a freeway, and relatively constant at 400 micrograms per cubic meter. That's not altogether easy, and Kostas can speak to that afterwards. But so we are actually been able to develop a dose response curve because we can't keep it at 400 micrograms, but we'll come back to that. Control animals are exposed to purified air. So what we are looking at is we simultaneously expose the animals to ovalbumin and concentrated air PM. So we're looking at adjuvant effects. We're looking at the enhancement of the immunologic sensitization by the particles. And we have done this at a site, as you can see, that is at close proximity to the 60 freeway, which is BH1. We have site 2, which is 150 meters from the freeway, which is site 2. And site 3 is 300 or 500? 500, yeah. And this is the prevailing wind. And so you see we get the effects of both the 5 freeway and the 60 freeway. And for those of you who've been there, these are heavily diesel traffic freeways. And so we are looking at freeways with a high percentage of diesel traffic as well as gasoline vehicles. And here are the results that we found. We found significant increases, statistically significant increases in IL-5 at 50 meters from the freeway. We find a statistically significant increase in IgG1 at 50 meters from the freeway. We find a significant increase in BH1 of eosinophils 50 meters from the freeway. And we find a significant increase in IL-13. So all the cytokines that I mentioned and immunoglobulins, we found statistically significant increases. And just to show you a picture, here we are at BH1 and BH2. And in this particular study, you'll notice that we found a stronger response in the fine plus ultrafine fraction. And in this case, we find the strongest response with IgG1 in the ultrafine fraction. Now, we asked ourselves, if ultrafines are the driving force for responses, I should go back and just point something out. Notice that we're talking about fine plus ultrafine. Our concentrator doesn't enable us to generate particles that don't include the ultrafine. So that when you see some of these results where we have fine plus ultrafine, keep in mind that you are seeing both the contribution from the ultrafine as well as the fine. That may be more detail than you wanted, but just so you're clear. So we plotted the IgG1, for example, versus particle number, and as you can see, we have a very strong correlation with particle and immunoglobulin formation. And there is an ultrafine point here that's covered up, but basically there's a very nice relation, even excluding, not excluding, but even recognizing this ultrafine point is off the charts. But we find a very strong relationship between particle number and immunoglobulin formation, which would tend to indicate that the small particles are generating the most significant effects. We find no relationship with mass concentration. So I'm going to shift now to continue the same animal studies. There have been speculations that neurologic disorders are associated with PM exposure, and there's some very wonderful work that's from the University of North Carolina on dogs associated with air pollution in Mexico City that exhibit lesions in brain tissue. And so we also collected brain tissue from the mice that were exposed for the allergic airway disease study, from BH2 in this case, and we've also now looked at BH1 with similar results. And we've looked at the expression of IL-1, TNF-alpha, and NF-kappa B, and these have all been reported in various studies to be inflammatory markers relevant to blood-brain barrier damage and CNS effects. And so we were following the literature when we chose these endpoints to look at. And as you can see, in the control animals, we get these results, and followed by the brains exposed to ultrafine and fine plus ultrafine, and we see increases in TNF-alpha, IL-1, and NF-kappa B. And so this is one study, but this is an interesting study given our mitochondrial work and given other important results in the literature. And this is something that we are going to follow up on, because it does appear that we're getting inflammatory responses in the brains associated with fine and ultrafine particle exposures. Finally, we've been frustrated trying to develop the best animal model, and so we looked at some geriatric rats. The geriatric rat is a potential model for particle effects in human populations, and we are particularly interested in potential cardiovascular effects in this regard. The problem for the experimentalists, I don't know what Kent's experience with this has been. I don't know if you've done geriatric rats. You have? The problem we have is that trouble with a geriatric rat is while we're holding them waiting to do the experiment, they tend to die. So there's always this time course that you're concerned about with respect to these studies. So this is a very preliminary study. It is designed to see if we thought this model was a model we wanted to follow up with. And without going through all the details, these are rats aged 24 to 26 months. They were exposed to the highest achievable fine plus ultrafines. Exposures were 4 hours per day for 3 consecutive days. There were 8 controls and 8 exposed rats. And we implanted 4 rats with blood pressure and EKG transponders, and they were exposed to fine plus ultrafine as well. The one problem we had with this particular study, besides the aging rats, is the 3 days that we did the study, something that almost never happens in Los Angeles occurred, namely it rained. So that when even though we're concentrating particles 40 times, the particle concentrations I think we were looking at were relatively low. Do you remember the exact numbers? Right. We would have preferred, since we had never done a geriatric rat experiment, we would have preferred to have 5 times that much. So we were running this experiment at 150 micrograms per cubic meter. So this is clearly not the model experiment. but we were under time constraints, and so we went ahead and did it. And we found that in the exposed animals, Mike Kleiman did see pre to post exposure changes in blood pressure. He did see heart rate changes in the animals, and so there were at least some indications of some cardiovascular alterations occurring. And he also looked at superoxide production in macrophages, and again he found in the fine plus ultrafine, he found increases in superoxide production. And I didn't put the slide up showing the cytokine measurements that he did. We also found significant increases in a wide number of cytokines, but that data is sufficiently preliminary that I didn't want to show it and create a false impression. So basically, we would conclude that we've seen significant inflammatory responses in brain tissue 2 weeks after CAPS exposure that have possible relevance to neurologic mechanisms. And we think that so far, with this extremely limited study, that the rat model was successful insofar as we did see changes in heart rate and macrophage responses. And so I've been talking for an hour. I'll stop. I just want to just one quick. This is our trailer at Rancho Los Amigos that was funded by the ARB. This is the duct leading into the concentrator, and this is the concentrator and the chambers for doing human clinical studies. And the reason I wanted to show you this is because I've spent this entire last hour trying to convince you that ultrafines are the most important compounds of interest from the standpoint of airway and cardiovascular effects. And so we obviously have an enormous amount of information associated with ultrafines and fine plus ultrafines. And so, but I wanted to end up by showing you that this whole business is relatively complicated. And so Henry Gong, in his human studies, where he's looking at healthy and asthmatic subjects, and this is the first clinical study that I'm aware of where human beings have been exposed to coarse particles, concentrated coarse particles. He saw no indication of airway effects. However, he did see increases in cardiovascular stress suggested by increased heart rate and decreased heart rate variability. So I want to end on that because I want to emphasize the fact that I think I would argue very strongly that ultrafine particles need to be taken seriously. The notion of particle number in a regulatory context, the notion of particle surface area in a regulatory context needs to be taken very seriously as opposed to simple standards. So we would argue about the importance of ultrafines, but in the end it's also clear that the problem is complex and that we're seeing effects from coarse particles as well. And so this is something that obviously needs to continue to sort out we would probably argue that reactive oxygen species, given our data, are not important elements of the toxicity of coarse particles. But that means something else is. And so I think that we'll continue to refine the reactive oxygen hypothesis and continue to do studies in that regard and look at issues of atmospheric chemistry and what have you. But that obviously issues of coarse particles and other mechanistic considerations becomes important. And so with that, I'll stop. Thanks.
That was great, John. The redox activity is certainly very important. There are many, many people who are studying that, and I'm wondering if you have any feelings for what we might anticipate if we were looking at redox activity as the potential culprit for health effects, adverse health effects, be it lungs or heart, when we add on to that ozone or other gaseous pollutants. Is there some interaction or potential synergism along those lines?
I think that's an absolutely crucial question that we don't have an answer to. I think that's a set of experiments that absolutely have to be done, and I don't know off the top of my head how to design them. Whether I think, for example, their DTT assay, what we have done, Kent, is we've mixed quinones with metals, and we get a synergistic response, that the metals seem to enhance the redox activity of the quinones. That's something I didn't want to report on because it's relative, but we haven't looked at the issue of putting in some form or capacity ozone with quinones or what have you to look at the redox activity. I think the experiment's not necessarily simple when you're dealing with a gas and a particle and vaporization and what have you.
Do you know when you use the concentrator, are you removing all ozone in that process, or is it maintained at ambient levels? Is there anything known about that?
Well, let me just say this, and Kostas can speak to it as well. For our in vitro samples, Kostas showed a slide, I think, this morning that shows you that we are collecting say the ultrafines with a liquid impinger, so that we're collecting something in liquid, so that to the degree that ozone is soluble in a liquid media, then there's going to be some ozone within that solution. How much we don't know, but so that, and one of the issues is we're interested in collecting things in liquid because that's more or less like the lung would be, but and because we have some questions about filter-based, what happens when you collect time on filters and the loss of potency that may be associated with that. But it is true that one can conceivably collect some soluble vapors within the impinger media. Is that fair, Kostas?
The concentrations of ozone actually are ambient. We don't enrich and we don't strip ozone. For that matter, I don't think we do this for any other gaseous pollutant.
Oh yeah, I'm sorry. I thought you knew that. Yeah. So
but
that
we
no gas. The gases aren't concentrated. It's only the particles that are concentrated.
But I don't think we are collecting effectively in the biosampler the impinger gases, in that the flow that we have, it's actually too fast for diffusion of gas molecules to get into the water. When you have 5 liters per minute, which to some degree, in a way, I wish it was collecting gases, because then we would have a great gas sampler, and most of the gaseous samplers are at 1/10 of those flow rates to be able to collect, to give adequate time to the molecules of any gaseous compound to be collected on a liquid surface. So in fact, we have measurements that we know that you're basically letting the ozone or nitrogen oxides, these are the 2 gaseous species for which we've done measurements, go in and out the biosampler, it's basically unaffected by the process. So it don't really collect those. But it would be, in a separate experiment, we could decrease the flow rate of our biosampler to the point that we can actually collect effectively, especially a soluble gas like ozone. So we could do that.
I think, just to add something to what you said, I think is extremely important. But I covered a lot of topics very quickly today, so it's silly to assimilate them all. But if you looked at those yeast data, there's other toxicity going on that doesn't have anything to do with reactive oxygen. So that obviously it's going to be a whole set of mechanisms that we need to be concerned about. But I would argue that reactive oxidative stress is certainly one of the more important ones. but it's not the only one by any stretch of the imagination. And so that's where, as you may appreciate, that our interest in capsaicin receptors arose, because we think other things are happening that are not fully explainable by reactive oxygen. And because Dane says that we're focused on oxygen too much, so we have to develop another half a dozen hypotheses to deal with it.
One thing that's always intrigued me, especially about diesel exhaust particulate matter, is that you have the expression of the carcinogenicity, plus you have these non-cancer effects. And does your work indicate what might be the tipping point? Is it simply a length of exposure that results in the carcinogenic effect, or is it something more basically mechanistic, where you'll have, all the redox reactions and the inflammation, whereas it's the epoxide route? But it's always been intriguing. This is cancer and non-cancer, and why one or the other, or maybe they're not exclusionary. It's just a curious thing I've wondered about.
Well, I think mechanistically they're different. I took out a slide that I wanted to show you that shows reactive oxygen reacting with DNA bases to produce adducts to DNA bases that people know very well produce G-to-tree transversions. And so one can certainly generate mutagenic responses from reactive oxygen. So there is a mechanistic pathway with oxidative stress towards carcinogenicity, as opposed to the more traditional view of the diol epoxide with BAP binding with the DNA, creating adducts, and so forth. And one of the things that's interesting about the diol epoxide, which everybody who took toxicology knows that pathway, is that pathway. The P53 gene is designed to take damaged DNA and prevent the S phase from occurring, and presumably the cell leading towards apoptosis. And so the degree to which you have a mutation in P53 that blocks that, then of course you have the sustained DNA damage that can go on to produce cancer. So it seems to me that I would argue at this point that the reactive oxygen pathway for carcinogenicity from a mechanistic standpoint is more important than I thought about it, say, 10 years ago. And that because when you start doing mutational spectra where you can actually see how a particular chemical metabolite affects a particular mutation, then you can begin to sort out different pathways. And I don't think I just answered your question, but I think that the redox activity is important. We've done experiments with our where we can produce DNA nicks and all sorts of things in DNA with reactive oxygen. It's a different seminar, but so that we think that there is an important pathway which has not been sufficiently explored. However, that pathway, the reactive oxygen pathway is the first step. It's the front end, right? Because that's where the hydroxyl radical or hydrogen peroxide or superoxide binds with something, either a sulfhydryl group or a DNA base or what have you. And subsequent, a whole series of then domino effects flow from that. And so emerges at the very end. We're answering questions at the very front end, but there's a whole pathway that we haven't answered very effectively. We and others haven't answered very effectively, and I think that's work yet to be done.
Just to follow up, John, do you think that the structure of the particle has anything to do with the endpoint? That is, you indicated that the organic fraction seemed to be the most responsible for the redox, but I remember earlier information that it probably was the carbon core that was responsible for the carcinogen. So I just wondered if there's any difference functionally in the structure.
Thank you very much, because I forgot something. That's the great question. Let me tell you what we have found. And when you make aluminum metal, you do it in aluminum smelters, and you use carbon as the electrode. So carbon clearly has some ability for electron transfer. You take bauxite, you take carbon coal tar electrodes, and you produce a lot of PAH, produce aluminum as well. Well, we've been interested in this notion of this idea that these particles have to pick up an electron from a reducing agent, so you have a way in which a quinone becomes a semiquinone, which then can react with oxygen. So we took some diesel exhaust particles and we did an electron spin resonance. And what we got knocked our socks off. We got, has everybody in here heard of the Buckminster fullerene molecule, the C60 molecule? That's this big ball. It's the buckyball, as it's called. We got that spectrum. We got the buckyball ESR spectrum. That's really amazing. And so we're going to write this up because it's actually fairly important. What the implication is that the carbon in diesel exhaust particles appears to have some structure which allows electron transfer. So that the carbon in the diesel appears to be paramagnetic. Well, what that suggests to us is that if you have this copper over here and this quinone over here, and you've got this soot in between, that actually the soot may function to transfer the electron from that copper to that quinone to produce a semiquinone. This is really oversimplification, right? So please. But it seems to me that the soot is not benign. I think the soot has electron transfer characteristics that we need to explore because we got this extraordinarily strong buckyball spectrum, which means that there's some impurity in there which has got to use the buckyball analogy, we've got some structure in it that has an electron sitting in the middle of it. And so the notion of transferring that electron is part of what we're talking about. So when I'm talking about when we're doing these electrochemical studies where we're actually measuring oxygen consumption, we're showing that there's an electrochemical process occurring on these particles that can literally reduce oxygen. We're measuring loss of oxygen from a particle, and so there has to be an electron transfer process. So that, I think that we are onto something that's really quite important in that respect. And again, but as I said earlier, it's a first phase, and all the other chemistry and biochemistry that occurs subsequent to that is obviously a complex process. And yeah, Dane? But let me just say, this electron spin resonance, it's just, you can't believe what we got. And why we got it, I don't know. I don't know.
Any possibility that explains why you might have axed in some of the cells that are being exposed because of the the electrochemical activity you can set up in there?
Possibly, but the thing that is interesting is what we've shown, and here's where a little humility helps, I think. What we've shown is that we can kill cells by destroying mitochondria. But that kills the cell. It doesn't produce asthma. It doesn't cause somebody to have a heart attack. It doesn't, it kills cells. And the trouble with killing cells is that they're no longer available to toxicity. So the whole issue, we've shown this wonderful uptake of particles into mitochondria, but there's a whole complex series of chemistries that we now need to work through, because what the toxicologic implications beyond cytotoxicity are an important issue. Because if the only thing we did was to kill cells, that would have a different level of significance, I think. Is that fair, Ken? I'm not the pathologist in the room, so I worry about being on a limb on this one. It is?
Okay, we have a couple questions off the web, yeah.
Yeah, it's been busy today. Request for research too. When you post the presentation material, could you also post the reference to the particulate matter study that you were talking about that dealt with effects on mitochondria?
Yes.
Okay. And there was a follow-up from Martin Rubin, earthlink.net, the person who wrote in about the jet fumes, and he was just recommending that you go east of the Santa Monica airport for your session. And then anybody else has a question? I think there's a couple of more.
COSIS, we can get COSIS to go east of the airport and we'll collect a sample and run a DTT sample and see how hot it is. We can certainly do that. But unless you're very close to a freeway, you don't get the same. The responses tend to be somewhat similar.
It's a long one. Okay, what effect would an incinerator fired at 1,800 degrees, burning 24 hours per day, 7 days per week, using diesel fuel of 4 plus or minus million gallons annually, natural gas and sulfur-laden clay at over 5,000 feet elevation with thin, dusty, and low cloud moisture and fractured or spherical particles within a 1,500 radius exposure to young children? I'm sorry if I didn't read that well.
It's okay. I'm going to just pass it to COSIS, who doesn't know the answer either.
Do you want me to email this to you?
I actually think that's a question for you folks and not for us folks. We're going to defer that.
Sounds like some vision, I think. Are there any more questions? Yeah, Andy.
Is your geriatric rat model just older individuals of a standard strain, or are you using one of these special engineered rats with one of the progeria genes in it?
Well, there are two answers to that. We had an opportunity to get some leftover rats free who were getting old. So, in terms of the carefully designed study, this was an opportunity of choice. So we took what we had, which were Sprague dollies, and they were 24 to 26 months. So we didn't spend a lot of time thinking about strains or even species. We just basically thought, let's go with it. We have them. Let's take a chance. The second point, however, that I feel very strongly about, we did a chronic animal bioassay on nutritionally deficient mice for arsenic, and we spent a lot of time thinking about what kind of knockout mouse might we choose. And the problem we came up with is that no matter what we thought, is that whatever knockout mouse or transgenic that we might, or rat, but whatever species or strain we had chosen would have been based on a specific hypothesis. So we had a P53 knockout, we would be thinking that P53 is important in arsenic carcinogenesis. So if we got a negative result, we would have, how do you decide whether that's relevant? So I think the selection of genetically altered species for studies like we're doing and others are doing, one really has to know what you're doing and what your hypothesis is and what your outcome may tell you if it's positive or negative. So I think that there's a lot of fishing expeditions that go on, and my feeling is that one has to be very careful if one is to get a meaningful result that doesn't just end up leaving you with more questions than you started with. So, no, but we're for example, since we have a reactive oxygen hypothesis, using a superoxide dismutase knockout was the perfect hypothesis. I mean, that worked. And of course, it's perfect because it did work, so,
I think with those experiments with transgenics is that if you get a positive result, you know something, but if you get a negative result, you don't know anything very much, or usually anyway.
Well, in our case, if you're doing a 2-year chronic study, you know you're not going to do it again in your lifetime, you want to try and you don't want to overly narrow your hypothesis, because you'll end up, if you get nothing, you'll be very frustrated, I think. So we went with a healthy strain, and we got a positive result, and so we think we understand that result. And therein lies some of the design problems, I think.
I think we'll end it there. We have another meeting after this. But John, I want to thank you for taking us from chemistry to biochemistry to toxicology, immunology, and back to toxicology again. So thank you very much to you and Costas for the presentations.
Great, thank you.